My interest is actually in tissue adhesion, so why tissues come together, why they seal up, how they can separate from each other, and in that portion, I needed an organ that closed. I went through a couple different options with where to go and decided on zebrafish first, and then had to find an organ that would open and close. And the more I learned about the eye, it had every aspect of research that I was interested in. Our zebrafish is see-through until about 5 days. So you can watch blood cells flow through veins, you can watch the heart beat, you can watch as the eye actually starts to come off of the brain during development. Most of our experiments we're trying to push towards a live imaging experiment. So zebrafish have the ability to have their genetics manipulated, which means we can insert genes that would lead to fluorescent proteins or to the glowing of certain areas of the fish. So we have a lot of transgenics, which is the traditional term, where we can have the eye glow green and the blood vessels glow red and the lens can glow a different color. And we can now color code and map the organ as we're watching it develop. We have been looking at a fish that we have the retina labeled in a green color. And when the eye develops, the way I like to describe it is if you had a balloon and you push the balloon should go over the actual lens, and it should go over in kind of a 360 turn. So it would make sense that it just pushes, but your eye actually forms like a baseball glove. So when the lens pushes in, the top part goes over, but the bottom part doesn't. It leaves this opening down at the bottom where the baseball glove would be open, and that's where your blood vessels come in and your retinal ganglion cells or your optic stalk, the optic nerve, will go out. And this all happens in a matter of about 24 hours in the fish. So what we found is while looking at these glowing fish is when it's closing, there's these cells that come off of the eye and they just kind of reach around. They reach around and touch the skin, they reach around and touch the lens, they reach around and touch each other. But they never do anything other than reach. And so what we're trying to figure out is, what are they doing? So I have this idea that they might be sensing the actual tissue and sensing the organism and figuring out positionally, where is my lens? Where is my skin? Where is my blood vessel? So that when the eye closes, it's going to close with the blood vessels in the back, with the optic nerve in the back, with the lens above it, and leave an opening for skin so that your retina doesn't fully encompass your actual lens and you have an opening for light to come through. So the one thing I did want to highlight is how much I really like UNC's emphasis on undergraduates getting involved in research. It is something that I knew I wanted as in my career as a faculty member. It was absolutely something that I looked for in any position that I was going to head towards. And seeing how excited students get and knowing that they're ready to come back, having them email me in summer, I got back a month early, when should I come in? It's just fun to watch them experience it because it was such a big part of my life and my trajectory that I'm excited to share that with other students.